Hello, friend. Welcome to Science for Sleep. Whether you are returning tonight or discovering this quiet place for the first time, this is a gentle corner to slow down, learn something peaceful, and let the day slowly fall away. Tonight, we are drifting into the soft, hidden world of bioluminescence. This topic was requested by a long-time, valued member of the channel, Dawn Taverner. The word bioluminescence simply means living light. It is the glow made by organisms themselves, not by fire, not by electricity in the way a lamp uses electricity, and not by reflected sunlight. It is light created inside a living body, through chemistry so delicate that a tiny insect, a drifting plankton cell, or a deep-sea fish can shine in the dark. This journey was inspired by a viewer request about David Attenborough's documentary Light on Earth. That film explored one of the strangest and most beautiful ideas in nature. All over the planet, in places most people rarely see, life has found ways to glow. In the ocean, blue sparks can flash through waves when tiny plankton are stirred by motion. In forests, fireflies can blink among leaves on warm summer nights. In caves, glowworm larvae can hang shining threads from stone ceilings, turning the dark above into something that looks almost like a sky. And far below the sea surface, where sunlight fades into black water, animals may use light to hunt, hide, warn, attract, or simply find one another. If you enjoy these quiet journeys, you're welcome to subscribe, and if it feels natural, to leave a comment sharing where you're listening from tonight. It helps others find their way to this calm corner of the world, one sleepy traveler at a time. The quiet continues here, just as it is. Bioluminescence can feel magical because it appears where light seems least expected. A black wave rolls toward shore, and for one brief moment, its edge shines blue. A beetle lifts from the grass, and its abdomen blinks green-yellow in the warm air. A piece of damp wood in an old forest gives off a faint green glow because certain fungi are quietly living inside it. These lights are soft, but they are not random. They come from real structures, real cells, and real chemical reactions. Many glowing organisms use a light-making molecule called luciferin. That name comes from an older word meaning light bringer. When luciferin reacts with oxygen, and when the right helper proteins are present, energy can be released as visible light. In many animals, very little of that energy becomes heat. That is why bioluminescence is sometimes called cold light. A firefly does not burn itself when it shines. A glowing plankton cell does not become a tiny flame. The light appears without smoke, without sparks, and without the harsh heat of fire. It is controlled from within the living body. For some creatures, the glow lasts only a fraction of a second. For others, it can pulse, blink, shimmer, or shine in patterns. Fireflies may flash in time signals to find mates. Some deep-sea fish carry glowing organs like small lanterns. Some squid can release clouds of glowing material into the water, leaving a bright distraction behind them as they slip away. And in some rare parts of the ocean, sailors have reported wide areas of steady pale glow, sometimes called milky seas, where the water seems softly lit across the surface. The beautiful part is that every one of these lights has a purpose. A glow can say, "Come closer." It can say, "Stay away." It can make a predator pause. It can make prey swim toward danger. It can erase a shadow from below, helping an animal blend into the faint light above it. In darkness, light becomes a kind of language. And because the dark parts of Earth are so large, that language is everywhere. The deep ocean covers most of the planet. Below a few hundred meters, sunlight grows weak. Below that, the water becomes a vast dim world, where ordinary vision is less useful, and even a small flash can carry meaning. A single blue-green pulse may travel farther than a body shape. A soft glow may reveal movement before an animal can be seen. A blinking pattern may be the difference between being alone and being found. So tonight, we will follow this living light slowly, from a shoreline that glows beneath moving waves, to the tiny chemistry inside a firefly, to deep water where animals carry lanterns in their skin, to caves, forests, and hidden places where life makes its own gentle brightness. And as the first small glow appears in the dark, there is no need to rush toward an answer. For now, it is enough to notice the light. That small light begins inside the body, long before it reaches the eye. Inside a firefly, the glow comes from a special part of the abdomen called the lantern. This lantern is made of thin layers of light-producing cells, with air tubes running through them like tiny breathing paths. When oxygen moves into those cells, the firefly can begin the chemical reaction that makes its familiar yellow-green flash. The main light-making molecule is called luciferin. Luciferin is the fuel of the glow. It stores energy in a form that can be released as visible light. Beside it is another helper called luciferase. Luciferase is an enzyme, which means it helps a chemical reaction happen faster and more neatly than it would on its own. In a firefly, luciferin, luciferase, oxygen, and a small energy-carrying molecule called ATP work together. ATP is the same basic energy molecule used by nearly every living cell, from human muscle cells to the cells inside a blade of grass. When these parts meet in the right order, the energy shifts. Instead of spreading out mostly as warmth, much of it leaves as light. That is why bioluminescence is often called cold light. A candle flame gives off heat because its energy is released by burning. A firefly's lantern stays cool enough for the insect's body because the reaction is controlled in living tissue. The glow appears softly, without smoke or flame. In warm summer fields, fireflies can turn this chemistry on and off with careful timing. Many species flash for less than a second at a time. Some male fireflies blink while flying above the grass. Females of the same species may answer from leaves or stems below. Each species has its own rhythm, so the flash pattern becomes a kind of address in the dark. One firefly may give a short burst. Another may wait a set number of seconds before answering. The chemistry is small, but the meaning can guide two insects toward each other across a field. The ocean uses the same broad idea on a much larger scale. Far below the surface, many animals do not use the exact firefly recipe, but they still depend on a light-making molecule, a helper protein, and oxygen. Some jellyfish use glowing proteins in their clear bodies. Some shrimp and squid release clouds of glowing liquid into the water. Some fish carry small light organs called photophores. A photophore is a living light spot, often built into the skin like a tiny lamp. In some deep-sea animals, these spots are arranged along the belly, around the eyes, or down the sides of the body. The placement matters. A light near the eye can help an animal search. A light on the belly can help hide its outline. A glowing lure near the mouth can draw prey closer. The same basic act, making light, can serve many different jobs depending on where the glow appears. Some creatures make their own light chemicals inside their bodies. Others depend on glowing bacteria. Flashlight fish, for example, carry bioluminescent bacteria in organs beneath their eyes. Those bacteria glow continuously, and the fish can cover or uncover the light, creating a blinking effect. The fish is not simply shining by accident. It is managing a living lamp. In the deep ocean, that kind of control matters. A glow that lasts too long can reveal an animal to predators. A flash at the right moment can confuse danger or help members of a group stay close. A hidden organ can become useful only when darkness asks for it. This careful control is one reason bioluminescence feels so different from ordinary light. A lamp shines because a switch sends power through a circuit. A glowing animal shines because its body moves molecules, oxygen, and energy through living cells. The result may be a tiny spark in a wave, a pulse from a jellyfish, or a soft lantern under the eye of a fish drifting through black water. Each glow begins with chemistry. Yet once that light enters the sea, the water itself changes what can be seen. Blue-green light travels farther through seawater than warmer colors. So the ocean slowly shapes living light into the color most likely to survive the dark. In seawater, color has to travel through a thick, moving body of liquid. Light that seems bright in air behaves differently once it enters the ocean. Sunlight is made of many wavelengths, which are tiny distances between waves of light. The colors humans see sit roughly between 400 and 700 nanometers. A nanometer is extremely small. 1 million nanometers would fit inside a single millimeter. Violet and blue light sit near the shorter end of that range. Orange and red light sit near the longer end. When sunlight enters clear ocean water, the longer red and orange wavelengths are absorbed quickly. Even a few meters below the surface, red objects begin to look duller. A red fish, a red diving glove, or a red coral branch can start to appear brown, grey, or almost black as the water above grows deeper. Blue and blue-green wavelengths travel farther because water absorbs them more slowly. That is why deep swimming pools often look blue, even when the tiles are white. The same rule shapes the open sea. Near the surface, sunlight still carries many colors. Lower down, the ocean slowly filters the sunlight until blue becomes the last strong color left. This matters for living light because a glow is only useful if another animal can see it. A red flash may be bright up close, but in open seawater it disappears quickly. A blue-green flash can travel farther through the same dark water. Many glowing marine animals produce light around blue or blue-green wavelengths, often close to 470 to 490 nanometers. That range passes through seawater well, especially in the clear open ocean far from muddy rivers and stirred-up coastlines. A tiny plankton cell glowing blue in a wave is making a color that the sea allows to move. A jellyfish pulse in dark water can be seen, Because its light matches the ocean's clearest window. A deep-sea fish with small glowing organs along its body often shines in the color most likely to reach nearby eyes. The water itself becomes part of the signal. It decides which colors fade and which colors remain visible. Coastal water can change the effect. Near shore, sand, silt, algae, and dissolved material can scatter and absorb light. After storms, a bay may look green or brown because particles are suspended in the water. In clearer tropical water, blue light can travel deeper. In colder or plankton-rich regions, green wavelengths may travel better than pure blue. So the color of bioluminescence is shaped by place as well as biology. The same glowing animal might stand out differently in a clear Pacific depth than in a cloudy coastal inlet. Some marine animals have evolved around these differences. Many deep-sea eyes are especially sensitive to blue-green light. Their retinas, the light-detecting tissue at the back of the eye, are tuned to the colors most common in their world. For them, a faint blue flash may carry as much meaning as a sound does in a quiet room. It can mark the movement of prey. It can reveal the edge of a body. It can show that another living creature is nearby. A few animals use rarer colors in special ways. Some dragonfish can produce red light from small organs near the eyes. Red light is unusual in the deep sea, and many animals there cannot see it well. This gives the dragonfish a strange advantage. It can shine a dim red beam over prey while remaining difficult for many other creatures to detect. The red glow works almost like a private searchlight in a world mostly tuned to blue. Even this exception shows how strongly the ocean controls color. Blue-green light becomes the common language because most eyes can receive it, and seawater can carry it. Red light becomes useful because many deep-sea creatures have lost the need to notice it. Every color has a cost. Every glow must match its surroundings. As the sea grows darker with depth, light becomes less like decoration and more like a precise tool. Near the surface, a bright flash may scatter through waves. Deeper down, a smaller glow can travel through still water with surprising clarity. Below a few hundred meters, sunlight weakens into a dim blue haze, and the creatures living there begin to depend on signals made by their own bodies. The ocean becomes a layered place, with daylight fading above and living lanterns appearing below. At about 200 meters below the ocean surface, the water enters a dim region scientists call the twilight zone. The name fits because sunlight still reaches this depth, but only as a weakened blue glow. At the surface, waves catch daylight. Clouds reflect on the water, and color still feels familiar. Lower down, the red and orange wavelengths have already faded. Shapes become softer. Edges lose detail. The sea turns into a vertical shadow, with brightness above and deep blackness waiting below. The twilight zone usually extends from about 200 meters to 1,000 meters deep. That is roughly the height of several stacked skyscrapers, measured downward through cold water. At 200 meters, there may still be enough light for some animals to sense day from night. At 1,000 meters, sunlight has nearly vanished. A human eye would find very little to hold on to there. Yet this layer is full of moving life. Tiny crustaceans drift through it. Gelatinous animals pulse slowly. Squid hover with careful control. Fish with large eyes watch the faint light above them. Many animals rise and sink through this zone each day in one of the largest movements of life on Earth. This daily movement is called vertical migration. Small animals often travel upward at night to feed closer to the surface, then sink deeper again before daylight returns. Some travel hundreds of meters in a single cycle. A shrimp-sized animal may move through more vertical distance, compared with its body size, than a person climbing a mountain every day. In this dim layer, bioluminescence becomes extremely common. Scientists estimate that around 80% of animals between 200 and 1,000 meters can produce light. Other deep-sea surveys have found that roughly 3/4 of animals drifting in the open water column are capable of glowing. That means living light is a normal part of the twilight zone. A flash there does not always mark something rare. It may be part of ordinary life, appearing as briefly as a blink, then fading before the water settles again. The reason is simple and powerful. In a world with little sunlight, even a small glow can change what happens next. A copepod, which is a tiny crustacean, may release a flash when disturbed. A jellyfish may pulse light along the rim of its bell. A squid may send a bright signal through skin cells or release glowing fluid into the water. A fish may carry rows of photophores, the tiny light organs that act like dots along its body. Each glow enters a world where eyes are waiting for faint signals. Some animals use light to be noticed. Others use it to avoid being noticed. Many seem to choose exactly when visibility helps them and when darkness protects them. This makes the twilight zone different from a simple dark room. It is more like a quiet city at night, where small lights appear for a purpose. A window glows for a moment. A signal blinks across a distance. A shape moves between patches of dim blue. The water is cold, often only a few degrees above freezing in many regions, and pressure rises quickly with depth. For every 10 meters of descent, pressure increases by about 1 atmosphere. At 1,000 meters, an animal is living under roughly 100 times the pressure felt at the surface. The creatures of this zone are built for that weight. Soft bodies, flexible tissues, and slow movements help many of them survive where human lungs and bones could never function unaided. Light gives these animals a way to act inside that pressure and darkness. A steady glow can soften a silhouette. A sudden flash can startle an attacker. A pattern can guide another member of the same species closer. In the open sea, where there are no trees, rocks, or cave walls nearby, a living signal may be the only landmark. The twilight zone is also connected to the planet's carbon cycle. When animals rise at night to feed near the surface, they eat carbon-rich plankton and carry some of that material downward again. Their waste, their molts, and their bodies help move carbon into deeper water. So the same layer filled with faint blue signals also plays a role in how the ocean stores energy from the surface world. The glow is beautiful, but it belongs to a working ecosystem. By the time the water reaches the lower edge of the twilight zone, living light has become part of hunting, hiding, feeding, mating, and drifting through the dark. A flash is no longer only a glow. It begins to feel like a message, sent through cold water by a body that has learned how to speak with light. That language can begin with bodies too small to see clearly in your hand. Many glowing shorelines come from dinoflagellates. Dinoflagellates are single-celled plankton, meaning each one is a complete living organism made of only one cell. Many are smaller than a grain of sand. Some are shaped like tiny spinning tops. Some carry two thin tails called flagella, which help them move through water with a slow turning motion. By day, many dinoflagellates drift near the surface, mixed among other plankton in sunlit water. Some species can use sunlight to make energy, much like plants do. By night, certain species become living sparks. They do not shine like steady lanterns. Their glow appears as brief flashes when the water around them is disturbed. A wave breaks. A paddle cuts through a bay. A swimmer moves an arm through dark water. For a split second, thousands or millions of tiny cells answer the motion with blue light. The result can look like blue fire rolling along the shore, even though the light is cool and chemical. Places famous for this glow include Mosquito Bay, on the island of Vieques in Puerto Rico, where dense populations of glowing dinoflagellates can make the water flash around boats at night. Similar displays appear in parts of Jamaica, the Maldives, Australia, California, and other warm coastal waters, when the right species gather in high numbers. The flash begins when the cell feels mechanical stress. Mechanical stress simply means pressure, bending, or movement pushing against the cell. Inside, some dinoflagellates are tiny light-producing structures called scintillons. A scintillon is like a small reaction chamber inside the cell. When the water moves sharply enough, chemical conditions inside the cell shift. Hydrogen ions, which are tiny charged particles, move in a way that changes acidity inside the light chamber. That change helps the luciferin and luciferase system release a burst of blue light. The flash may last only about 1/10 of a second. Yet when countless cells flash together, the water seems to ignite beneath the surface. This glow may help protect the plankton. Small grazers, such as copepods, feed on plankton. A copepod is a tiny crustacean, related in a distant way to shrimp and crabs. When a copepod bumps into glowing dinoflagellates, the sudden flash can startle it. The light may also reveal the grazer to larger animals, such as small fish. This idea is sometimes called the burglar alarm effect. The plankton's flash can draw attention to the creature that disturbed it. In a dark sea, that moment of attention may change who gets eaten. The same effect can become visible on a much larger animal. In David Attenborough's Light on Earth, glowing plankton reveal moving shapes in black water. A dolphin swimming at night can become outlined in blue sparks. The animal itself does not have to glow. Its body presses through the water, and the plankton around it flash in response. For a few seconds, the dolphin's path appears as a bright moving sketch. Its nose, fins, and tail leave trails of living light. Fish can carve thin blue lines through the dark. A whale rising near the surface can stir a broad glow along its sides, as if the sea is drawing the animal only after it has already moved. This is one reason bioluminescence feels so strange to human eyes. The light can show motion more clearly than shape. A wave may be invisible until it breaks. A hand may be hidden until it moves. A passing animal may appear as a glowing outline, then vanish as the cells fade back into darkness. The plankton are not trying to make the ocean beautiful for us. They are reacting to pressure, motion, and danger in the only world they know. Even a gentle ripple can become information. Even a footstep in wet sand can wake thousands of flashes. On a quiet beach, each blue spark is the response of a single living cell. Together, those cells turn movement into a visible trail. The shoreline becomes a place where every wave writes briefly in light. Most of these flashes disappear almost as soon as they appear. The water darkens again. The surface smooths. The glowing cells continue drifting with the tide. Yet the ocean holds other forms of light that behave differently. Some do not vanish after a single touch. Some spread outward across the surface until whole areas of sea seem to shine from within. At the larger scale, living light can become wide enough for a ship to sail through. For centuries, sailors crossing the Indian Ocean and parts of the Arabian Sea described nights when the water seemed to turn pale around them. The surface did not sparkle in quick flashes. It shone with a broad, steady glow, sometimes white, sometimes faintly green, spreading toward the horizon like moonlight trapped under the waves. These events became known as milky seas. In old ship logs, sailors wrote about water that looked like snow, clouds, or glowing milk. The reports sounded almost impossible because ordinary bioluminescent plankton usually flash when disturbed, then fade almost at once. A milky sea behaves differently. It can appear calm. It can cover huge distances. Some accounts describe light lasting for hours as a vessel moves through it. Others suggest the glow may remain across the same ocean region for days or even weeks. One famous modern report came from the merchant ship Lima in 1995. The ship was traveling in the northwestern Indian Ocean when the crew reported entering a glowing area of sea. The water looked pale from horizon to horizon. The ship's wake, normally brighter when plankton flash, appeared almost dark against the surrounding glow, because the whole surface was already shining. Years later, scientists compared the ship's recorded position with satellite data from that night. They found a large glowing patch in the same region, captured by a low-light satellite sensor. The estimated area was enormous, roughly the size of a small country. That discovery helped turn old sailor stories into a real scientific target. Milky seas are still rare and poorly understood, but one leading idea involves luminous bacteria. One species often discussed is Vibrio harveyi. This bacterium can produce a soft blue-green light when many cells gather together. The process depends on something called quorum sensing. Quorum sensing is a way for bacteria to sense crowd size. Each cell releases tiny chemical signals into the water. When only a few bacteria are present, the signals spread out and stay weak. When the population becomes dense, the signals build up. Once enough signals are detected, many bacteria can switch on their light-producing genes at the same time. A single glowing bacterium would be far too small for human eyes to notice. A vast number of them, spread through the surface water, could create a steady glow. This may explain why milky seas look different from sparkling plankton displays. The light comes from a huge population acting together, instead of individual flashes triggered by movement. Scientists think these bacteria may gather on floating organic material, algae, or tiny particles rich in nutrients. The ocean surface is never empty. It carries oils, decaying plankton, bits of mucus, drifting cells, and microscopic debris. For bacteria, that material can become a floating meal. If conditions are calm enough, and if enough nutrients gather in one place, bacterial numbers may rise until their chemical signals reach the level needed for collective glow. The result could be a softly lit skin across the water. From the deck of a ship, the scene would feel dreamlike. The bow would push into pale water. The railings would sit above a sea that seemed quietly illuminated from below. No bright sparks would be needed. No breaking wave would have to strike the hull. The glow would already be there, stretching out in every direction. Satellite detection changed how scientists think about these reports. Human witnesses are rare because ships must happen to cross the right place at the right time. Aircraft may pass too high or too quickly. Modern satellites can look across entire ocean basins at night, searching for faint light patterns that last longer than a passing flash. Even with satellites, milky seas remain difficult to confirm because moonlight, clouds, storms, and sensor limits can confuse the picture. A true milky sea has to be separated from reflected light. Ordinary plankton flashes and other bright features on the ocean surface. That careful work makes the mystery quieter, but also stronger. Milky seas show that bioluminescence can belong to whole drifting communities, not only single animals or single cells responding to touch. The ocean can become a shared glowing field, shaped by bacteria, nutrients, currents, and time. A chemical signal too small to see can grow into a light visible from space. Once living light reaches that scale, it becomes more than a strange ocean effect. It begins to reveal how survival can depend on when a creature shines, how long the signal lasts, and who else is watching in the dark. A watcher in the dark may be a predator, a mate, a rival, or a passing animal with no clear intention yet. That is why living light can never be understood by brightness alone. The meaning depends on the body making it, the place where it appears, and the creature that receives it. A flash from plankton near the surface may mark danger. A steady glow under a fish may help hide its shape. A blinking pattern from a firefly may guide a mate through summer grass. In the ocean, this language is especially rich because darkness gives light more power. A small signal can stand out across open water. A soft pulse can travel through blue-black space where smells spread slowly, and sound may come from many directions at once. For many animals, bioluminescence is part of daily survival, as real as fins, eyes, shells, or teeth. Deep-sea squids can use light in several ways. Some carry tiny photophores in their skin, arranged like dots along the body. These light organs may help break up the animal's outline, so it blends into the dim water around it. Other squid can release glowing fluid into the sea when threatened. The cloud hangs behind them like a bright, false target. A predator may turn toward that shining patch while the squid moves away into darker water. The glow buys time. In the deep sea, even a few seconds can matter. Jellyfish use light with a different kind of softness. Many have clear bodies, thin bells, and long trailing tentacles. A disturbance can send a pulse of light through the jelly's rim or along its body. Some flashes may startle an attacker. Others may draw the attention of larger hunters nearby. That creates a risk for the animal that first touched the jelly. A small predator may suddenly become visible to something bigger. The light changes the balance around it. Fish have turned bioluminescence into lanterns, lures, and patterns. Some deep-sea anglerfish carry a glowing lure that hangs near the mouth. The lure may contain bacteria that produce light inside a small, fleshy bulb. In the black water, that moving point can look like an easy meal. A smaller fish comes closer, following the glow until it reaches the hidden mouth behind it. The anglerfish does not need to chase across long distances. Its light lets hunger come to it. Other fish use rows of glowing organs along the sides of the body. These rows can help members of the same species recognize each other. In open water, where there are no branches, stones, or reef walls nearby, a pattern of dots may become an identity mark. A fish can signal that it belongs to the same group. A group can stay together while drifting through the dark. Crustaceans add still another form of light. Some small shrimp-like animals release glowing chemicals into the water when disturbed. The glow spreads outward like a pale cloud. To a predator, the sudden brightness can be confusing. To a larger hunter, it can mark the location of movement. The animal that produced the light may slip away, while attention gathers around the shining cloud. Even marine worms and corals can glow. Some deep-sea worms flash when touched or attacked. Certain corals and relatives of corals can give off light when disturbed, sending faint blue or green signals through their soft tissues. These animals often live slowly, attached to surfaces or drifting with little speed. For them, light may be a defense they can use without running. A glow can make contact more costly for whatever disturbed them. This is why bioluminescence can feel like a language, even though it has no words. One signal can invite. One signal can warn. One signal can mislead. The same color may carry a different meaning depending on where it appears. A blue dot near a mouth may be a trap. A blue flash from plankton may expose a grazer. A blue glow along a belly may help an animal avoid being outlined from below. Darkness gives each signal room to work. The water becomes a quiet field of possible meanings. A pulse appears, travels, and fades. Nearby eyes may read it as food, danger, company, or cover. For a creature living there, the glow is never just beautiful. It belongs to the hard, careful work of staying alive. And among all these uses, the most delicate trick is the one that almost hides itself completely. Some animals make light so their own bodies become harder to see. Seen from below, even a small animal can become a dark mark against the faint glow above. A predator looking upward through deep water is often searching for silhouettes. The body of a fish or squid can block the last traces of sunlight filtering down from the surface. That blocked patch becomes a shape, a meal-sized shadow, a clue. Counter-illumination is the name for one of the ocean's quietest camouflage tricks. The word means lighting from the opposite side. An animal produces light on its underside, usually from small organs called photophores, so its belly blends with the dim light coming from above. Photophores can look like tiny dots, cups, tubes, or patches in the skin. Some are packed with light-making chemicals. Some contain glowing bacteria. Some are connected to nerves and muscles that help the animal adjust brightness. The goal is careful matching. Too little light leaves a shadow. Too much light creates a glow that stands out. The animal survives by finding the narrow middle, where its underside becomes close to the brightness of the water behind it. In the twilight zone, this can matter every hour of the day. At 300 or 500 meters deep, the surface may still send down a weak blue haze. To an animal swimming below, anything above can appear as a dark outline against that haze. A hatchetfish, for example, has a thin, flat body and rows of photophores along its lower side. These fish often live in the mesopelagic zone, the dim middle layer of the sea. Their bellies can shine downward just enough to soften their outline. From beneath, the fish becomes less like a sharp shape and more like part of the water itself. Some squid use this same principle. The Hawaiian bobtail squid, a small species often only a few centimeters long, carries bioluminescent bacteria in a special light organ. It hides in sand during the day and comes out at night to hunt in shallow coastal water. Moonlight and starlight can create a shadow beneath it. By glowing from its underside, the squid can reduce that shadow while it moves above the seafloor. Its bacteria, called Vibrio fischeri, live inside the squid's light organ and help produce the glow. The squid provides shelter and nutrients. The bacteria provide light. Together, they form a living camouflage system. Some deeper squid have more complex light organs arranged across the body. Their glow may be adjusted by filters, reflectors, and tiny shutters in the skin. These structures can change the way light spreads. A reflector can direct light outward. A filter can shift color. A shutter can dim or block the glow. The animal is not simply shining. It is shaping light with living tissue. Counter-illumination also depends on direction. A fish seen from the side may still be visible. A squid seen from above may blend into darker water. The trick works best for a predator looking upward, where the bright background gives away the prey's shape. That is why so many photophores are placed on the underside. Their position follows the direction of danger. The light answers a specific kind of seeing. This kind of camouflage shows how deeply the ocean has shaped life. On land, animals often hide with color patterns that match bark, leaves, soil, or snow. In the open water, there may be no surface to match. There is only light above, darkness below, and empty space around the body. A glowing belly becomes the background. A living body becomes part of the dim blue field. The same glow that could reveal one creature can conceal another. That depends on brightness, angle, timing, and the eyes watching from below. A soft row of photophores along a fish may erase a shadow. A sudden flash from a jellyfish may draw attention. A glowing lure may bring prey closer. Light carries many meanings because darkness creates many needs. Counter-illumination is one of the most delicate meanings because success is measured by being missed. No chase begins. No attack is launched. No outline is noticed in time. Somewhere under a fading blue ceiling of water, a small animal passes through the view of a hunter and remains almost part of the sea. Yet camouflage also creates pressure. When one creature learns to disappear with light, another creature may evolve better eyes, sharper filters, and new ways to read the glow. Better vision changes the meaning of every flash. In the deep sea, eyes are shaped by a world where light is rare, faint, and often made by other animals. A bright eye is useless without enough light to enter it. So many deep-sea creatures have enlarged eyes, wide pupils, and sensitive retinas. The retina is the layer at the back of the eye that receives light and turns it into nerve signals. In humans, the retina works well in daylight because our eyes are built for a bright world. In the twilight zone, an eye may need to catch a flash that lasts less than a second, or a dim blue glow moving far away through water. That pressure has created some unusual forms of vision. One of the strangest belongs to the cockeyed squid, also known as the strawberry squid. Its scientific name is Histiotuthis heteropsis. It gets the strawberry nickname from its reddish body, covered in small pale spots, which can look like seeds on fruit. But its eyes are the truly remarkable part. One eye grows much larger than the other. The large eye is usually yellowish and points upward. It watches the faint blue light filtering down from the surface. That upward-looking eye can help detect silhouettes, such as the shadow of a fish, shrimp, or squid, passing above. The smaller eye points more downward and outward. It is better placed to notice bioluminescent flashes in the darker water below. The animal lives between two visual worlds. Above it, there is weak sunlight. Below it, there are sparks from living bodies. The squid's face carries both directions at once, with one eye searching the dim ceiling of the ocean and the other watching the scattered lights beneath it. This is a quiet example of evolution working with available light. The squid does not need matching eyes if the two sides of its world offer different information. One eye gathers the last traces of daylight. The other reads the glow of animals hidden below. Its body becomes a small drifting observatory in the water, slowly turning through darkness with two different tools for seeing. Other deep-sea animals solve the problem in their own ways. Some fish have tubular eyes, shaped more like narrow barrels than round balls. These eyes point upward and gather light from a small part of the water above. The barrel-eye fish is one famous example. Its transparent head allows its greenish tubular eyes to look upward through a clear dome of tissue. Those eyes can search for silhouettes against the faint light overhead, while the fish remains below potential prey. Some animals have yellow or red filters in their eyes. A filter is tissue that changes which colors reach the retina. In a blue-green world, filters can help separate ordinary background glow from a signal that matters. Certain deep-sea fishes may use these filters to notice specific types of bioluminescence more clearly. The eye is no longer a simple receiver. It becomes a sorting system, deciding which light deserves attention. This matters because the ocean's living light can be deceptive. A glow may be a lure near the mouth of an anglerfish. A flash may be a plankton alarm. A soft belly glow may be camouflage. An animal that can read the difference gains a better chance of surviving. The same blue-green color can carry several meanings. so the eye must judge motion, shape, brightness, direction, and timing. A steady row of dots may belong to a fish moving calmly. A sudden cloud may mark a frightened shrimp or squid. A pulse along a jellyfish may warn of contact. The deep sea is filled with signals that appear briefly and disappear before a slow eye could understand them. Vision there must be sensitive, patient, and quick enough when the moment comes. Large eyes also come with costs. They take energy to build. They take space in the head. They can make an animal more noticeable. So when an animal has unusually large or specialized eyes, it tells us that seeing faint light is worth the cost. The darkness has shaped both sides of the exchange. Bodies learn to glow. Eyes learn to interpret the glow. Camouflage became more precise. Detection became more careful. Across millions of years, this back and forth created a quiet arms race in the water. A hidden animal shines just enough to disappear. A hunter watches for the small mismatch. A lure glows near a mouth. A cautious eye studies whether the light moves like food or like danger. In this world, sight is not only about brightness. It is about reading tiny differences inside darkness. And some creatures have taken that reading further by carrying their own lamps, often with the help of living bacteria tucked safely inside their bodies. Those lamps can be shared between very different forms of life. In flashlight fish, the glow does not come only from the fish's own cells. It comes from bacteria living inside special light organs below the eyes. Flashlight fish belong to a small family of fishes called Anomalopidae. Many live around coral reefs and rocky slopes in warm parts of the Indo-Pacific, including waters near Indonesia, the Philippines, and the Red Sea. At night, they can rise from darker reef cracks and move in groups through black water, each fish carrying a pair of glowing patches on its face. The light organs sit like small blue-green windows beneath the eyes. Inside them are dense colonies of bioluminescent bacteria. The bacteria receive shelter, oxygen, and nutrients from the fish. The fish receives steady light from the bacteria. This kind of relationship is called symbiosis. Symbiosis means two different organisms living closely together in a way that affects them both. In this case, the partnership is so close that the fish has built body parts around the bacteria's glow. The bacteria shine continuously. The fish controls what other animals can see. In some flashlight fish, the light organ can be covered by a dark fold of skin, almost like closing a tiny eyelid. In others, the organ can rotate or tuck into a pocket, so the light disappears from view. This creates a blinking effect. A fish can flash, hide the glow, then flash again as it swims. In darkness, that pattern becomes useful. A group of flashlight fish can stay together by watching one another's signals. A quick burst of light may help a neighbor keep position. A sudden blackout may help the group scatter when danger passes nearby. In reef water at night, when coral branches and rock ledges become dark shapes, a blinking cheek lamp can work like a quiet signal between bodies moving close together. These lights can also help with feeding. Small plankton and tiny crustaceans often gather in the water after sunset. A flashlight fish can use its glow to search, giving itself a small circle of vision beneath the reef. The light may also attract little animals close enough to catch. For a creature with a small mouth and quick movements, that glow can turn darkness into a hunting space. The same idea appears in a more dramatic form in deep-sea anglerfish. Many female anglerfish carry a modified fin spine that extends forward from the head. At the end is a soft lure called an esker. The esker can glow, often with help from bioluminescent bacteria living inside it. In deep water, a moving point of light can resemble prey. A small fish or shrimp may approach the lure, drawn by the only bright object nearby. The anglerfish waits behind it, with a wide mouth and long teeth set in shadow. The lure makes distance shorter. Instead of searching through miles of dark water, the predator lets the signal work around its body. This is one of the great differences between a lantern and a lure. A lantern helps an animal see or be recognized. A lure changes another animal's behavior. Both depend on attention. Both use darkness as part of the design. A light that would vanish in daylight becomes powerful in black water because the background is almost empty. Bacterial light also shows how evolution can borrow. An animal may not need to invent every chemical tool by itself. It can create a safe place for microbes that already glow. Over many generations, that partnership can become part of the animal's shape, habits, and survival. The fish grows the organ. The bacteria provide the shine. The ocean receives a signal made by two lives working as one. These glowing partnerships remind us that bioluminescence is rarely a simple decoration. A blink beneath the eye can hold a school together. A glow near the mouth can guide prey closer. A covered light can help a fish vanish for a moment, then return as a soft blue point in the dark. Once light can guide, gather, and lure, it can also change the fate of an attack. A sudden flash may call attention from far away, turning one predator's secret movement into a signal for something larger nearby. That larger presence can change everything in a place where attacks often happen in silence. In the deep sea, a predator may approach through black water, with almost no warning. There may be no reef to hide behind. There may be no sunlight to reveal a shape from far away. For some animals, the best defense is a sudden burst of light. One of the most famous examples is the Attenborough jellyfish. Attenborough lives in deep ocean water around the world, often hundreds or thousands of meters below the surface. Its body is shaped like a small reddish umbrella, with a bell that may measure several centimeters across. Around the edge of that bell are thin tentacles, and one longer trailing tentacle can stretch behind it as it drifts. In ordinary deep-sea darkness, its red color is difficult for many animals to see. Red light fades quickly underwater, so a red body can become nearly hidden at depth. Yet when Attenborough is disturbed, it can produce a bright blue-green display. The light travels around the rim of the bell in a moving ring. Sometimes the pattern looks like a spinning wheel. Sometimes it appears as a circle of blue points pulsing outward through the dark. This display has given Attenborough the nickname alarm jelly. The name comes from one of the most interesting ideas in bioluminescence. A flash may protect the animal by drawing attention to the attacker. If a smaller predator grabs the jellyfish lights up. That glow may be noticed by a larger predator nearby. The larger animal may approach the light and chase, bite, or disturb the original attacker. In that way, the jellyfish's signal can turn danger back toward the creature causing it. Scientists sometimes call this the burglar alarm effect. The image is easy to understand. A hidden intruder becomes safer in darkness. A bright alarm makes the intruder visible. The animal producing the alarm may get a chance to escape during the confusion. This idea has been tested in deep-sea exploration in a very creative way. Researchers built an optical lure called an jelly. It was designed to imitate the flashing display of a toller. The lure was attached to a deep-sea camera system known as Medusa. Medusa was created to observe deep animals with less disturbance than bright research lights or noisy vehicles might cause. Instead of flooding the water with harsh light, the system could wait quietly and use a glowing pattern that deep-sea animals might already recognize. The jelly produced circular blue flashes like a living alarm signal. That artificial glow helped attract large deep-sea predators close enough to film. One of the most famous results was footage of a giant squid in its natural deep-water world. For centuries, giant squid were known mostly from dead bodies washed ashore, pieces found in whale stomachs, and rare encounters near the surface. Seeing one alive in deep water required a signal that made sense in its environment. The jelly worked because it borrowed a language already present in the sea. A toller's light also shows how defense can be active without speed. A jellyfish cannot sprint away like a fish. It drifts, pulses, and moves with slow contractions of its bell. Its soft body offers little armor. A bright alarm gives it another option. The flash can startle. The moving ring can distract. The glow can call attention from beyond the reach of touch. Other deep-sea animals use similar defensive tricks in different forms. Some shrimp release glowing clouds into the water. Some squid leave bright decoys behind them. Some worms flash when grabbed, creating a sudden signal at the point of attack. These lights can interrupt the simple path between predator and prey. A hunter expects darkness. A flash changes the scene. A glowing cloud may mark the wrong target. A pulsing jelly may make the attacker visible. A sudden burst may give a soft-bodied animal just enough time to drift away. In the deep ocean, survival often depends on timing measured in seconds. A light that lasts briefly can still redirect attention. A signal that fades quickly can still change a decision. A toller's blue ring is beautiful to human eyes. Yet beauty is only part of what we notice from the outside. For the jellyfish, that circle of light belongs to pressure, contact, and escape. It is a calm-looking signal with serious work inside it. Down in the dark water, living light has become a strategy. It can lure, hide, warn, and call. Far above that deep pressure, on warm land after sunset, another kind of glow rises from grass and trees with a quieter rhythm, timed through the bodies of small beetles blinking in the night. On summer evenings, the glow moves through air instead of water. A small light rises above the grass. It blinks once, then disappears. A few seconds later, another light answers from a leaf or low stem. To human eyes, the scene can feel peaceful and simple, but each flash belongs to a careful pattern. Despite their name, fireflies are beetles. They belong to a family of insects called Lampyridae. A name that comes from older words linked with shining. There are more than 2,000 known firefly species around the world, and many of them use light to communicate after sunset. Their glow comes from a lantern in the abdomen, where luciferin, luciferase oxygen, and ATP work together in a controlled chemical reaction. The same basic idea we followed earlier appears again on land, inside a body light enough to rest on a blade of grass. For many fireflies, the glow is part of courtship. Males often fly above the ground, flashing as they move. Females may wait lower in the vegetation, watching from leaves, stems, or the edges of fields. When a female recognizes the right pattern from a male of her species, she may answer with her own flash. That reply can guide the male toward her through the dark. The timing matters. One species may flash quickly. Another may hold the glow longer. Some flash close to the ground, while others rise higher among trees. A firefly searching for a mate has to send the right signal and read the right answer. The dark becomes a quiet conversation made of timed light. In the Great Smoky Mountains of Tennessee and North Carolina, one species has become famous for turning this private signal into a shared display. Its name is Photinus carolinus. For a few weeks each year, usually from late May into June, males gather in large numbers and flash in loose synchrony. They often produce a burst of about 5 to 8 flashes, then leave several seconds of darkness, often around 8 seconds before the pattern begins again. The result can make a forest slope appear to breathe with light. The trees brighten in scattered points. The ground falls dark. Then the flashes return together, softly enough that the whole scene still feels quiet. The timing does not begin perfectly. Individual males adjust to the flashes around them. One insect blinks. Nearby insects shift their rhythm. Slowly, a shared pulse forms across the forest. This is called synchronous flashing. Synchronous means happening at the same time. The behavior likely helps females identify the correct species more easily, because the pattern becomes clearer when many males follow it together. A single flash in a crowded forest could be missed. A coordinated rhythm stands out against the night. Photinus carolinus is only one example. Other firefly species have their own flash grammar. Some produce quick single pulses. Some trace a short glowing path as they fly. Some females answer after a precise delay, like a quiet signal sent back through leaves. In certain species, females from one group can imitate the flash pattern of another species. Those false replies may lure males close enough to be eaten. Even on land, living light can carry attraction and danger inside the same glow. Firefly light also changes with the environment. Warm evenings usually bring more activity because insects are cold-blooded and their body processes depend partly on outside temperature. Moist habitats often support strong firefly populations, since many firefly larvae live in damp soil, leaf litter, or near water. The larvae can glow too. In some species, immature fireflies shine from the ground, where they hunt small snails, worms, and soft-bodied prey. Their light may warn predators that they taste bad or contain defensive chemicals. So the firefly's glow is not only a signal for love. Across its life, it can help protect, identify, attract, and survive. A child watching a field at dusk may simply see tiny lanterns drifting in warm air. Underneath that calm view, each light belongs to a specific body, a species pattern, and a chemical system older than human language. The beetles write their signals in short strokes of green-yellow light, then let the darkness return between them. Beyond fields and forests, another land glow waits in deeper shadow. In caves, some small larvae use light in a slower and more patient way, turning ceilings into star-like traps for insects that fly too close. Inside a limestone cave, darkness can feel almost complete. The walls hold cool moisture. Drops of water gather on stone. Sound moves gently through narrow chambers and returns as a soft echo. Then, above the path or above a slow underground stream, tiny blue-green lights appear across the ceiling. They can look like stars. Each point of light belongs to a small larva waiting for food. In New Zealand, the famous glowworm species is Arachnocampa luminosa. Despite the name glowworm, this animal is the larval stage of a fungus gnat. A larva is the young form of an insect before it changes into its adult shape. A caterpillar is the larva of a butterfly. A glowworm is the larva of a small fly-like insect. Arachnocampa luminosa lives in dark, damp places, especially caves, overhangs, stream banks, and shaded forest areas where the air stays moist. Moisture matters because the larva's hunting threads can dry out in open air. Inside caves, the stable humidity helps those threads remain sticky. The glow comes from the rear end of the larva's body. The light is usually blue-green, a color that stands out strongly in cave darkness. Flying insects inside the cave may move toward that glow, the way they might move toward moonlight or a gap in the trees outside. Above them, the larva waits in a small silk hammock attached to the cave roof. From that hammock, it lowers thin fishing lines. These lines are made of silk and coated with sticky droplets of mucus. Some can hang several centimeters long. In dense colonies, hundreds or thousands of threads may dangle from the ceiling like clear beaded curtains. A tiny fly, moth, or midge rises toward the glow and brushes against one of the lines. The sticky droplets hold it. The larva then pulls the thread upward and eats the trapped insect. The scene looks peaceful from below, but it is a quiet form of hunting. The ceiling shines because each larva is advertising a trap. The Waitomo glowworm caves on New Zealand's North Island are one of the best known places to see this. The caves are made of limestone, a rock formed from ancient marine shells and skeletons pressed together over long stretches of time. Water slowly dissolves limestone, carving tunnels, chambers, and underground passages. At Waitomo, visitors have been guided through the caves since the late 1800s, giving the site more than 130 years of tourism history. Boats move quietly along dark water while the ceiling glows overhead. The stillness helps protect the colony. Loud noise, touching the cave walls, strong lights, and changes in airflow can disturb the animals or the damp conditions they need. The glowworm life cycle also gives the display a special timing. The larval stage is the long feeding stage. This is when the animal glows, builds threads, and gathers enough energy to transform. Later, it becomes a pupa. the resting stage where the body changes. Then it emerges as an adult fungus gnat. The adult form lives only briefly compared with the larva and does very little feeding. Its main purpose is to mate and begin the cycle again. So the cave lights are mostly the lights of young insects gathering energy before transformation. That makes the scene even stranger and more delicate. A ceiling that looks like a night sky is also a nursery, a hunting ground, and a hidden insect community. Each glow is small, each thread is thin. Together, they create one of the most beautiful displays of bioluminescence on land. David Attenborough's work helped bring scenes like this to a wider audience, because filming such faint light requires patience and sensitive cameras. Ordinary bright lamps can overwhelm the glow. A camera has to wait with the darkness, letting the blue-green points gather slowly in the image. The cave then reveals itself without being forced. Living light has now moved from waves to deep water, to forest fields, to stone ceilings. It can shine in open ocean and in enclosed darkness. It can flash through plankton or hang above a cave stream. And beneath the forest outside, another kind of glow can come from old wood, wet leaves, and the quiet work of fungi breaking matter down. Under a damp forest canopy, light can come from places that seem almost asleep. A fallen branch rests beneath brown leaves. Rainwater darkens the bark. Moss grows along one side. Inside the wood, fungal threads are slowly spreading through the softened fibers. On certain nights, when the air is humid and the forest floor stays dark, that old branch may give off a faint green glow. This glow has a long old name: foxfire. For centuries, people used the word foxfire for pale light shining from rotting wood in forests and swamps. Before electric lamps, travelers sometimes noticed it along dark trails. Soldiers and miners were said to use pieces of glowing wood as a weak natural marker. The light was never bright like a torch. It was soft enough to feel secret, as if the wood itself were remembering daylight. Today, we know that foxfire usually comes from fungi. Fungi are living organisms that include mushrooms, molds, yeasts, and the hidden thread-like networks that grow through soil and wood. The visible mushroom is often just the fruiting body. Much of the fungus lives as mycelium. Mycelium is a web of tiny threads, called hyphae, growing through dead wood, leaf litter, or soil. Those threads release enzymes, which are chemicals that break large materials into smaller pieces. In a rotting log, fungal enzymes help break down cellulose and lignin. Cellulose is the tough material that gives plant cell walls their structure. Lignin is the harder material that helps wood stay stiff. Together, they make a tree strong while it is alive. After the tree falls, fungi help return that stored material to the forest. Some glowing fungi shine from the mushroom cap. Others glow from the gills under the cap, or from the mycelium hidden inside wood. One famous glowing mushroom is Omphallotus olearius, sometimes called the jack-o'-lantern mushroom. It grows in clusters near tree roots or on buried wood, and its gills can glow faint green in darkness. Another group, Armillaria, includes honey fungi that can produce glowing mycelium inside decaying wood. In tropical Brazil, Neonothopanus gardneri can glow strongly enough that its large mushrooms are visible at night in the forest. Its pale green light rises from the cap and gills like a small, quiet lamp among leaves. Fungal bioluminescence uses its own chemical system. It still depends on oxygen, and it still releases energy as visible light. Researchers have found that glowing fungi use a light-producing molecule different from the one in fireflies. The reaction is linked to compounds made from caffeic acid, a natural chemical found in many plants and fungi. This means a mushroom's glow has a different recipe from an insect's flash, even though both create living light. The forest version is usually slower and steadier. A firefly may blink. A plankton cell may flash when touched. A glowing fungus may shine for hours, while its cells continue their quiet work inside damp wood. The reason fungi glow is still being studied. One idea is that the light attracts insects. In a dark forest, a small green glow may guide beetles, flies, or other tiny animals toward the mushroom. When they visit, they may carry fungal spores away on their bodies. Spores are the tiny reproductive cells fungi use to spread, somewhat like seeds, though much smaller and simpler. If insects help move those spores, the glow may help the fungus reach new logs, new soil, or new patches of leaf litter. Another idea is that the glow may be connected to the fungus's internal chemistry. Breaking down wood involves complex reactions with oxygen. Some of the same chemical pathways that manage those reactions may also produce light. The glow may carry more than one purpose, shaped by both survival and chemistry. A glowing log is part of a larger cycle. The fallen tree once lifted water through its trunk and held leaves in the sun. After it falls, beetles tunnel through it. Fungi enter cracks in the bark. Bacteria gather in the damp spaces. The wood softens, opens, and slowly becomes food for the forest floor. In that slow return, a small green light may appear. Bioluminescence on land is much rarer than in the sea, which makes these fungal glows feel especially quiet. They do not fill whole ocean layers. They appear in hidden places, close to the ground, often after rain. Yet they carry the same deeper pattern. Life finds darkness, then uses chemistry to send a signal through it. In the forest, that signal may help insects find a mushroom. In the ocean, it may help a fish hide, hunt, or answer another body nearby. Living light has now appeared in beetles, bacteria, plankton, jellyfish, squid, fish, larvae, and fungi. The same result keeps returning in different branches of life, each one arriving by its own path through evolution. Across the tree of life, this repeated glow tells a deeper story. Bioluminescence has appeared many times in separate groups of organisms. A beetle in a meadow, a jellyfish in deep water, a bacterium on drifting particles, and a mushroom on a wet log can all make light, but they do not all use the same chemical recipe. The broad result looks similar. The pathways inside the body can be very different. This pattern is called convergent evolution. Convergent evolution means separate living things arrive at a similar solution because they face similar problems. A bat and a bird both fly. Though their wings came from different ancestors, a shark and a dolphin both have smooth bodies shaped for swimming, though one is a fish and the other is a mammal. Living light follows that same kind of pattern. different branches of life entered darkness, then found ways to make brightness useful. Fireflies use firefly luciferin. Many jellyfish and other marine animals use a molecule called coenterazine. Some bacteria use a system involving a molecule related to vitamin B2, along with a long-chain chemical called an aldehyde. Glowing fungi use a pathway linked to histidine, a compound connected to the chemistry of plant and fungal tissues. Each system has its own parts. Each one needs oxygen. Each one releases energy as visible light. The details vary because evolution works with whatever materials are already present in a body. A useful chemical reaction may begin as a way to manage oxygen or remove unstable molecules. Over time, if the reaction gives off light, and that light helps survival, the glow can become sharper, brighter, and more controlled. A faint byproduct can become a signal. A weak shine can become a lure. A small reaction can become an organ, a pattern, or a behavior. In the ocean, darkness creates many chances for light to become useful. Deep water covers most of Earth's living space. Below the bright surface layer, animals still need to find food, avoid predators, and locate mates. A flash can travel farther than a body scent in open water. A glow can appear exactly when a creature wants attention. A hidden light organ can disappear behind skin, then shine again when needed. Because the ocean is so large, even a small advantage can matter across millions of generations. This helps explain why bioluminescence is far more common in the sea than on land. Land has darkness too, but it also has branches, leaves, soil, burrows, and many surfaces where animals can hide by shape or color. The open ocean offers fewer hiding places. A body surrounded by water has only distance, movement, and light to work with. So light becomes part of the landscape. It becomes a tool shaped by hunger, fear, mating, and camouflage. The pressures are different in each place. A firefly flashing over grass needs a mate of the same species to recognize its timing. A glowworm larva in a cave needs flying insects to move toward its sticky threads. A dinoflagellate near the surface may benefit when a flash exposes the grazer that disturbed it. A hatchetfish in the twilight zone needs its belly lights to match the dim glow overhead. A fungus on rotting wood may gain visitors that help move its spores through the forest. The reason for glowing changes, yet the advantage always depends on darkness making light meaningful. This repeated evolution also reminds us that bioluminescence is older and more widespread than any single familiar example. Fireflies are only one chapter. Glowing beaches are only one chapter. The deep ocean holds a much larger library of living light, written in bodies we rarely see alive. Some of those bodies are soft and clear. Some are red and nearly invisible at depth. Some are no larger than a grain of rice. Some drift through water thousands of meters below the surface, where pressure is heavy enough to crush ordinary equipment. Scientists still have to untangle which glows share ancient roots and which appeared separately. They study genes, molecules, body structures, and behavior. A gene is a set of instructions in DNA that helps a body build proteins. By comparing genes from glowing organisms, researchers can see whether two light systems are closely related or only look similar from the outside. The more they look, the more living light seems like a set of many inventions arranged across the planet. Each glow belongs to its own history. Each history begins with a body facing darkness and finding a way to use it. To understand those histories, human eyes need help. The faintest glows are brief, soft, and easily lost. So scientists and filmmakers have had to build better ways of seeing what the dark has been holding all along. That help often begins with a lens pointed patiently into darkness. Bioluminescence is difficult to film because many glows are faint, brief, and easily drowned out by ordinary light. A firefly in a field can be seen by the human eye because the background is dark and the insect is close. A tiny jellyfish hundreds of meters underwater is much harder to capture. Its light may last for only a moment. Its body may be nearly transparent. Its signal may vanish if a bright lamp shines directly into the water. For a long time, this made living light hard to study. Scientists could collect animals in nets, bring them to the surface, and examine them in tanks. That helped reveal body parts, chemistry, and species names. Yet it often missed the animal's real behavior. A deep-sea creature lifted from cold, high-pressure water may arrive damaged, stressed, or already changed by the journey. The glow seen in a lab can be useful, but the glow in its natural darkness tells a richer story. This is why better cameras changed the way people understood bioluminescence. David Attenborough's Light on Earth helped show how much of this world depended on new filming tools. The documentary used highly sensitive cameras, custom lighting systems, and underwater technology to reveal glows that older cameras would have missed. Some of the low-light cameras used in modern nature filming can detect extremely small amounts of light, far beyond what older equipment could capture. A camera described as thousands of times more sensitive than one from a decade earlier can turn a nearly invisible glow into a visible image. That does not mean the light becomes brighter in nature. It means the instrument has become better at waiting for weak signals. The camera gathers photons. A photon is a tiny packet of light. When only a few photons arrive from a glowing animal, the camera must collect them carefully, reduce background noise, and hold the image steady enough for the pattern to appear. Noise, in camera language, means random speckles or false signals that can cover fine detail. Low-light filming is partly a battle between real faint light and electronic grain. In the deep ocean, robots and submersibles expanded the search. A submersible is a small underwater craft built to carry people or instruments down into deep water. A remotely operated vehicle, often called an ROV, is controlled from a ship through a cable. These machines can descend hundreds or thousands of meters while carrying cameras, sensors, sampling arms, and lights. They allow scientists to watch animals where they actually live, floating in the cold pressure of the water column. Still, the machines have to be used carefully. Bright white lights can scare animals away or change their behavior. The sound and movement of a vehicle can disturb the water. A fragile jellyfish may drift out of view before its glow appears. A squid may flash only once, then vanish into blackness beyond the camera. For this reason, some deep-sea systems use dim red light, low-light cameras, or special lures that mimic natural bioluminescence. Many deep-sea animals are less sensitive to red light, so red illumination can sometimes help researchers see without overwhelming the scene. Other systems wait in darkness and record only when movement or a glow appears. The goal is to let the deep sea behave like itself. The collaboration between scientists and filmmakers matters here. Scientists know the species, the depths, the likely behaviors, and the careful limits of observation. Filmmakers know how to capture faint scenes in a way that human eyes can understand. Together, they can reveal a dolphin outlined by glowing plankton, a cave ceiling covered in blue-green larvae, or a deep-sea animal pulsing once in front of a silent camera. The image may feel effortless when viewed on a screen. Behind it are long hours of waiting, careful equipment, and darkness protected from too much human light. A camera staring into black water may show nothing at first. Then a small body drifts close. A faint pulse opens along its edge. For a second, the animal becomes visible by its own chemistry. The screen holds what the eye alone would have missed. Better cameras reveal the glow, but careful methods reveal the behavior behind it. To understand living light, scientists must enter darkness gently, because the wrong kind of brightness can erase the very world they came to see. The deep sea has to be approached almost like a sleeping room. Too much light changes the scene. Too much movement sends animals away. A loud machine, a bright beam, or a collecting net can turn natural behavior into a reaction to human arrival. For bioluminescence, that problem is especially important. Many glowing animals shine only when touched, startled, chased, or pressed by moving water. A flash recorded in front of a camera may be a natural signal. It may also be a response to the camera itself. Scientists have to separate those two possibilities with care. Remotely operated vehicles have helped more than almost any other tool. An ROV is an underwater robot controlled from a ship through a long cable. The cable carries power, instructions, and video signals. Some ROVs can descend thousands of meters, where pressure is hundreds of times stronger than at the surface. They carry cameras, lights, robotic arms, temperature sensors, water samplers, and sometimes gentle collection containers for fragile animals. Institutes that study the deep sea have used ROVs for decades to watch animals in place. This has revealed jellyfish, comb jellies, squid, crustaceans, worms, and fishes drifting in the water column, with far more detail than old net samples could show. The water column simply means the open water between the surface and the seafloor. It is not a wall or a place with edges. It is the great vertical space through which animals drift, swim, rise, and sink. In that space, many soft-bodied creatures are easily damaged by nets. A jellyfish can tear. A comb jelly can collapse into clear fragments. A delicate siphonophore, which is a colonial animal made of many connected parts, can break before it ever reaches a ship deck. ROVs allow these animals to be seen before the shape is lost. Even so, an ROV brings its own disturbance. Its thrusters push water. Its metal body casts a shape. Its lights can be far brighter than anything normally present at 700 or 1,000 meters deep. A deep-sea animal that spends its life in faint blue darkness may respond strongly to a sudden white beam. It may stop moving, it may flash. It may turn away or drift out of view. For this reason, researchers often use softer methods when they can. Low-light cameras can record faint glows without flooding the scene. Red light can sometimes help, because many deep-sea animals have limited sensitivity to red wavelengths. Stationary camera platforms can sit quietly and wait, letting animals come near without the constant motion of a vehicle. Baited cameras may attract scavengers. Optical lures may mimic natural glow patterns. Each method changes the scene a little, but careful design can reduce the disturbance. The hardest part is patience. A camera may watch dark water for hours. Most of the frame may stay empty. Tiny particles drift past like dust in a moonbeam. A transparent animal may arrive so slowly that it seems to form out of the water itself. Then a touch, a current, or a small collision can trigger one brief flash. That moment has to be captured without turning the entire habitat into a stage. Deep-sea research also depends on knowing depth, time, temperature, and location. A glow seen at 300 meters may have a different meaning from one seen at 1,500 meters. A flash near the surface at night may come from an animal migrating upward to feed. A flash in deeper water during the day may belong to a creature that never sees sunlight at all. Researchers build records from many observations, slowly mapping where glowing animals live and how often they appear. Over time, those records show patterns. Some groups are common in the twilight zone. Some are linked to deeper black water. Some flash only during contact. Some carry steady organs that glow as part of daily life. Studying bioluminescence means studying both the light and the silence around it. The absence of a glow can matter too. An animal may hold its light back until danger is close. A fish may cover its lamp. A squid may stay dark until the exact second it needs a decoy. The behavior is often hidden in timing. That is why careful observation has become so valuable. The more gently scientists enter the dark, the more the dark can show its ordinary life. And once living light could be measured, filmed, and understood with more care, it began to move from the ocean into laboratories, where the same chemistry became a tool for seeing hidden processes inside cells. Inside a laboratory, living light becomes small enough to fit in a clear plastic dish. Rows of cells may grow in a thin layer of liquid. To the unaided eye, they look almost blank. A pale surface, a quiet sample, a small container resting under a hood, or inside an incubator. Yet inside those cells, genes may be switching on, energy may be rising or falling, and tiny changes may be happening long before anything visible appears. Bioluminescence gives scientists a way to see some of that hidden activity. One of the most useful tools comes from fireflies. Firefly luciferase is the enzyme that helps firefly luciferin produce light. Because the reaction is bright, sensitive, and easy to measure, researchers have used it for decades in molecular biology. Molecular biology is the study of the tiny instructions and processes inside living cells. A cell contains DNA, which acts like a library of instructions. Genes are sections of DNA that tell a cell how to build certain proteins. When a gene is active, scientists say it is being expressed. Gene expression simply means the cell is using that instruction. The clever part is that scientists can attach the luciferase instruction to another gene they want to study. If that gene becomes active, the cell also makes luciferase. Then, when luciferin is added, the cell gives off light. The glow becomes a visible sign that something inside the cell has turned on. It is like placing a tiny lamp beside a hidden switch. When the switch is active, the lamp shines. This kind of system is called a reporter system. The luciferase acts as the reporter because it reports what is happening inside the cell. A camera or light measuring instrument can detect how strong the glow is. A brighter signal can mean more activity. A weaker signal can mean less activity. The result is gentle to imagine, but powerful in practice. A dark room, a plate of cells, a faint glow appearing where a process is active. Researchers use this approach to study how genes respond to medicines, stress, infection, hormones, nutrients, or changes in the environment around the cell. The glow does not require the cell to be cut open each time. In many experiments, scientists can watch changes over time. A signal may rise after a treatment is added. It may fade as a process slows. It may appear in one group of cells and stay absent in another. Bioluminescence turns invisible timing into something measurable. Firefly light is also useful because it depends on ATP. ATP is the small energy-carrying molecule used throughout life. Earlier, we met it as part of the firefly's glow. In the lab, that same reaction can help measure how much ATP is present in a sample. When luciferin and luciferase are added, more ATP can produce more light. Because living cells need ATP, this gives researchers a way to estimate cell activity, cell number, or contamination. For example, food safety labs and medical labs can use ATP-based tests to detect tiny traces of living material on surfaces or in samples. The glow becomes a quick sign that biological activity is present. The idea is quiet and precise. A molecule from a beetle's lantern helps reveal energy inside places too small to see. A chemical signal that once guided insects through grass now helps researchers measure cells in glass dishes. This is one reason bioluminescence has had such a large scientific life beyond nature documentaries and night beaches. It is beautiful because it shines. It is useful because the shining can be counted. Instruments can measure light in tiny amounts. They can compare one sample with another. They can follow changes minute by minute. A human eye may only see a faint glow, while a machine can record exact values and turn them into data. Bioluminescence also has advantages because most cells and tissues do not naturally glow strongly in the same way. That makes the signal easier to notice against a dark background. A small amount of light can stand out clearly when the surroundings are quiet. The same principle works in the ocean. A blue flash matters because the water around it is dark. In a lab, a glowing sample matters because the instrument is waiting in darkness too. Living light has become a bridge between wonder and measurement. It belongs to fireflies, plankton, jellyfish, bacteria, fungi, and deep-sea fish. It also belongs to microscopes, cameras, incubators, and careful hands working in quiet rooms. Yet the more useful this light becomes to science, the more it reminds us that nature still holds many versions of it we have only partly understood. Many glows can be measured. Many others still appear briefly in the dark, carrying meanings that remain unfinished. Those unfinished meanings are part of what makes bioluminescence so difficult to fully explain. Scientists understand the basic outline. A molecule stores energy. Oxygen helps release it. A protein guides the reaction. Light appears. Yet across the living world, that outline changes again and again. A firefly's lantern, a glowing bacterium, a deep-sea jellyfish, and a green shining mushroom may all produce light, while using different chemical parts to do it. Some organisms make their own luciferin inside their bodies. Others seem to gain light-making molecules from the food they eat. Many marine animals use a luciferin called coenterazine, which appears in jellyfish, shrimp, squid, fish, and other ocean creatures. The strange part is that not every animal using it seems able to build it from scratch. Some may collect it through the food web. A small animal eats glowing prey. A larger animal eats that small animal. Over time, the light-making molecule may move through bodies, like a hidden ingredient passed through the sea. This creates a quiet puzzle. If an animal glows, the chemistry inside it may be its own invention, a borrowed tool, or a partnership with microbes. A flashlight fish carries bacteria. A firefly builds its own lantern system. A mushroom uses a fungal pathway tied to its own metabolism. A deep-sea animal may rely on molecules gathered from meals eaten in darkness. The glow looks simple from outside. Inside, it may come from a long chain of life, diet, genes, organs, and chance. Even when the chemistry is known, the purpose can remain uncertain. Some signals are fairly clear. A firefly flash helps mates find each other. A glowworm cave light draws insects towards sticky threads. An anglerfish lure hangs near a mouth in deep water. A row of belly lights can help erase a shadow. These examples connect light to behavior that can be watched and tested. Other glows are harder to read. A jellyfish may flash when touched, though the exact receiver of that signal may depend on which predators are nearby. A deep-sea worm may release light during contact, while the animal that benefits from the flash may never be filmed. A squid may glow in patterns that change with body posture, depth, or fear. A soft drifting creature may pulse once in front of a camera, then leave no second chance to compare. The deep sea makes these mysteries harder because many animals live far from easy observation. At 1,000 meters, pressure is about 100 times stronger than at the surface. At 3,000 meters, it is roughly 300 times stronger. Temperatures can hover near freezing. Many animals are delicate enough to be damaged by nets, bright lights, or the trip upward. A species may be known from only a handful of sightings. Some are filmed alive only once or twice. Others are collected without ever showing their natural glow. This leaves scientists with fragments. A flash recorded by an ROV, a body examined on a ship, a molecule tested in a lab, a gene sequence compared with relatives. Each fragment helps, while the full behavior may remain hidden in black water. There are also evolutionary puzzles. bioluminescence has appeared many times, which means the advantage must be strong in the right conditions. Still, the first step is difficult to reconstruct. A weak chemical glow may have begun as a side effect of oxygen chemistry. If that faint light helped even slightly, natural selection could shape it over many generations. Brighter cells could become useful. Useful cells could gather into organs. Organs could gain reflectors, lenses, covers, or filters. A small accidental shine could slowly become a controlled signal. That path sounds reasonable, yet each lineage may have followed a different route. Bacteria, fungi, beetles, jellyfish, and fish do not share one simple origin story for light. They carry separate histories written into their chemistry. A single unknown flash in the deep ocean can hold all of that uncertainty. It appears in the camera frame, blue against black water. It widens for a moment, then fades before the animal fully comes into view. The signal may have warned, hidden, lured, startled, or answered something outside the frame. Human tools can record the light. The meaning may still belong to the dark space around it. That is why living light remains both known and unfinished. The science is real and growing. The molecules have names. The depths can be measured. The flashes can be filmed. Yet the largest glowing habitat on Earth is still the deep ocean, and much of that world remains only partly seen. Across that hidden volume, the scale of living light becomes hard to picture. The ocean covers about 71% of Earth's surface. Its average depth is close to 3,700 meters. Most of that space lies below the bright surface layer, where sunlight can power coral reefs, kelp forests, and the floating plant-like plankton that drift near the top. Below about 200 meters, daylight weakens into the twilight zone. Below about 1,000 meters, the bathypelagic zone begins, a deep layer where sunlight no longer guides ordinary vision. That zone stretches down to around 4,000 meters in many parts of the ocean. Farther still, trenches can descend past 10,000 meters, deeper than Mount Everest is tall. All of this water makes the open ocean the largest living space on the planet. A forest has trunks, branches, soil, roots, and clear surfaces to rest on. A reef has coral heads, cracks, ledges, and sandy patches. The open water column has fewer fixed places. It is a vast vertical habitat made of pressure, temperature, movement, and distance. Animals there live suspended in layers. Some drift almost like glass ornaments. Some pulse slowly through the water. Some swim with narrow fins and large eyes. Many carry light. Long-term deep-sea observations have suggested that more than 3/4 of animals drifting in the water column can produce bioluminescence. That means living light may be one of the most common traits in Earth's largest habitat. The idea changes the way the deep sea feels. From above, the ocean can look like a dark, flat surface at night. From inside, it is layered with signals. A copepod may release a tiny flash when disturbed. A comb jelly may ripple with blue-green light along its rows of moving cilia. Cilia are tiny hair-like structures that beat in waves, helping the animal move. A siphonophore may stretch through the water as a long, delicate colony, with parts specialized for floating, feeding, and defense. Some siphonophores can be longer than a school bus, yet their bodies are so fragile they can break apart in rough collection nets. A squid may carry photophores in careful patterns. A fish may hide its outline with belly lights. A jellyfish may pulse a blue ring when touched. Together, these signals make the deep ocean more patterned than it first appears. There is motion in the dark. There are warnings in the dark. There are quiet displays that last only long enough for nearby eyes to receive them. The light is often brief, because brightness has a cost. Making light takes energy. Revealing light at the wrong time can draw predators. A creature that glows constantly must have a reason, such as hiding a shadow, finding food, or keeping contact with others. Many animals keep their glow under control, saving it for a moment when darkness becomes useful. This makes the deep ocean feel less like a black, empty space and more like a low, hidden conversation. Most of the signals never reach human eyes. A flash may happen 800 meters below a calm surface, while a ship passes above with no sign of it. A glowing animal may drift through the Monterey Canyon, the Mid-Atlantic, or the waters near Japan, sending a signal that fades before any camera arrives. A fish may blink beneath a layer of water thick enough to hold back nearly every color of sunlight. The deep sea also changes slowly compared with the world above. Temperatures in many deep regions stay cold and steady. Food often arrives as marine snow, the soft fall of dead plankton, waste, mucus, and tiny particles sinking from the surface. This sinking material can feed animals far below the sunlit zone. In that slow fall of food, animals hunt, hide, and signal with light. The glow belongs to the same system as feeding and migration. It is part of how life organizes itself, where landmarks are scarce. A person standing on a beach at night may see only black water beyond the waves. Beneath that darkness, the largest habitat on Earth may be filled with small controlled lights, appearing one by one through cold layers of water. The reason this world feels hidden is partly because human life is built around sunlight. Our eyes expect surfaces, colors, and steady shapes. The deep ocean speaks in dim flashes, brief outlines, and signals that vanish almost as soon as they arrive. Our senses are tuned for a brighter world. Human eyes work beautifully in daylight, where surfaces hold color, edges stay clear, and movement can be followed across open space. The retina has two main kinds of light-detecting cells. Cones help us see color and find detail when light is strong. Rods help us see in dim conditions, though they give us less color and less sharpness. After entering darkness, the eyes slowly become more sensitive. This process is called dark adaptation. It can take around 20 to 30 minutes for human night vision to improve strongly after bright light fades. Even then, our eyes are still limited. A faint glow from plankton may be easy to miss if a phone screen has just been shining nearby. A soft fungal glow on a fallen branch may vanish beneath a flashlight beam. A glowworm cave may need silence, darkness, and patience before the ceiling begins to look like a field of small blue stars. Bioluminescence often sits close to the edge of what we can notice. Some of it is too brief. Some of it is too faint. Some of it happens too far below us, under hundreds of meters of water. A dinoflagellate flash may last only a fraction of a second. A deep-sea jellyfish may pulse once when touched by a current or a passing animal. A fish may cover its light organ before a camera comes close. The event happens, leaves its signal, and settles back into darkness. This is one reason living light can feel like a secret. It is real, measurable, and chemical, yet it often appears only under the right conditions. A person walking along a beach might see nothing unusual at first. Then a wave folds over itself, and the foam glows blue for a moment. A foot presses into wet sand, and tiny sparks appear around the edge. A hand moves through black water, and the trail shines behind the fingers. The light was already there in the organisms. Motion gave it a reason to show itself. Other forms of bioluminescence need stillness instead. In a cave, bright lamps can wash out the glow from larvae overhead. When the light is lowered, the ceiling slowly gathers points of blue-green. In a damp forest, the glow from fungi may be hidden under leaves, inside softened wood, or along the mycelium spreading through a fallen log. A person might walk past it many times in daylight and never know the branch could shine after dark. The world changes when the usual light is removed. A beach becomes a surface that can flash. A cave becomes a sky. A rotting log becomes a quiet lamp. The deep ocean takes this further than any familiar place. A ship may cross above thousands of glowing animals without seeing a single one. Below the hull, in layers of cold water, small bodies may be flashing, hiding, hunting, and drifting. Their signals do not rise to the surface. Their messages stay inside the water column, where the right eyes can receive them. Human life is arranged around sunlight, lamps, screens, streets, rooms, and windows. We move through places made bright enough for our eyes to work quickly. Bioluminescence belongs to slower looking. It asks for darkness to remain dark. It asks for a wave to move, a beetle to blink, a mushroom to shine from wet wood, or a deep-sea animal to drift close enough for one faint pulse to appear. That makes it a gentle reminder about perception. The visible world is only the part our bodies are ready to receive. Around that visible world are signals too soft, too fast, too small, or too deeply hidden for ordinary attention. Living light does not need to be large to matter. A single cell can flash in disturbed water. A larva can glow from a cave roof. A bacterium can join millions of others and help turn the sea pale. A squid can use a small belly light to erase its own shadow. These quiet glows show that darkness is filled with activity, even when it looks empty from above. As the journey settles, the lights begin to gather into one simple shape. Chemistry, color, depth, motion, signals, cameras, and hidden habitats all lead back to the same soft truth. Earth has always held more light than daylight alone can show. All through this quiet journey, the same soft pattern has appeared in many forms. A living body gathers oxygen, a light-making molecule, and a helper protein. In fireflies, that helper is luciferase, working with luciferin inside the lantern of the abdomen. In many ocean animals, other luciferins and glowing proteins create blue-green flashes in water. In bacteria, whole colonies can shine together when enough cells gather and chemical signals build around them. In fungi, the glow can rise from damp wood, thread-like mycelium, or pale mushroom gills. Each version has its own recipe. The shared result is a small release of energy as visible light. That light often carries very little heat, which is why a firefly can flash without burning, and a plankton cell can glow without becoming a spark. The ocean shaped much of this story. Blue-green light travels especially well through seawater, so many marine animals shine in colors near that range. Red and orange fade quickly with depth, while blue can move farther through clear water. That simple fact helps explain why so many glowing beaches, jellyfish pulses, fish lanterns, and plankton trails appear blue to human eyes. The sea chooses which colors can travel, and life answers in the colors the water allows. Between about 200 and 1,000 meters deep, the twilight zone becomes one of Earth's great living light habitats. Sunlight weakens there, yet it does not vanish all at once. Animals drift between the last faint glow from above and the darker layers below. In that dim space, a large share of animals can produce light. For some, the glow attracts a mate. For others, it lures prey closer, warns of danger, or startles an attacker. Some fish and squid use counter-illumination, shining from the belly to soften the shadows seen from below. A glow can help a body become part of the faint blue water around it. Tiny plankton can turn motion into blue flashes along a shoreline. A wave, a paddle, a swimmer, or a dolphin can disturb the cells, and the water briefly draws the movement in light. Milky seas show the larger scale, where glowing bacteria may spread across broad regions of ocean and create a steady, pale shine. A taller jellyfish can send a blue ring through the dark when disturbed. Flashlight fish can carry glowing bacteria beneath the eyes, like living cheek lamps. Anglerfish can hold a lure in front of the mouth, letting darkness make one point of light irresistible. On land, fireflies show that bioluminescence can become a timed grammar. A male flashes over grass. A female may answer from leaves below. In the Great Smoky Mountains, Photinus carolinus can gather in large groups, blinking several times, then resting in darkness before the rhythm returns. In New Zealand caves, Arachnocampa luminosa larvae hang sticky threads from stone ceilings, using their glow to draw insects upward. On forest floors, foxfire can shine from old wood as fungi break fallen trees back into the soil. David Attenborough's Light on Earth helped open a doorway into this hidden world, showing how sensitive cameras, patient filming, and careful science can reveal glows that ordinary eyes often miss. Better tools showed that living light was never confined to one strange corner of nature. It was waiting in waves, caves, forests, reefs, deep water, laboratories, and drifting bodies that rarely meet the surface. Still, many meanings remain unfinished. Some animals make their own luciferin. Some may gather light-making molecules from food. Some carry bacteria that shine for them. Some flashes are easy to connect with hunting or mating, while others appear once in black water and leave their purpose unclear. That uncertainty feels fitting for a topic made of faint signals. Bioluminescence teaches through smallness. A single cell can flash when touched by a wave. A beetle can blink above a field. A mushroom can glow beneath wet leaves. A fish can hide its shadow with carefully measured light. A jellyfish can turn contact into a silent alarm. The lights are gentle, yet each one belongs to survival, chemistry, timing, and place. As the journey settles, the planet feels a little less empty in the dark. The black ocean holds drifting signals. The cave roof holds patient blue points. The forest floor holds a green shine inside old wood. Living light does not need to fill the night to change it. A single glow is enough to show that darkness is active, inhabited, and quietly alive. So let the last image be simple. One small blue-green point appears in deep water. It shines for a moment, soft and controlled. Then it fades back into the surrounding dark, leaving the darkness whole, with the light resting gently inside it.